This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV. This Week in Virology, episode 1241, recorded on August 1st, 2025. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Austin, Texas, Rich Condit.
Hi there, Vincent. How you doing?
All right. Haven't seen you in a while.
No, I've been goofing off.
You've been in Bend.
I've been in Bend, Oregon, or nearby, and it's been very, very nice. I got to kind of reboot from, believe it or not, being just too wound up in stuff, even though I'm retired. This is crazy. But there you go. Actually, most of the stuff I'm wound up with has to do with singing, so I can't complain. All right. The weather here in Austin, Texas is 100 degrees, 100 Fahrenheit, and we have mostly blue skies, a few puffy clouds. That is normal for this time of year, pretty much, and I'm not complaining because July was unusually mild. We had only one 100-degree day. We've had only 4, maybe 5 so far this season, so that's a break. And the reservoirs are full, and I had a break in Bend, so it's all good.
Here in Chelsea, New York, it is 23 C after a month of 34. Yesterday, a thunderstorm rolled through, and that's it. August 1st, boom, crap weather. There you go. Also joining us from Montreal, Canada, Angela Mingarelli.
Hello, everyone. It is also 23C, which I believe is 73F in Montreal. Vincent and I were talking about that this morning. It was fairly cold for August 1st. It was 12C when I woke up this morning. Hopefully the rest of August is not like that because, as many people know, Montreal winter is already long enough. So we want all the heat we can. Rich, send us some of your heat, please.
Sure thing.
Also joining us from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody. Here it's 76 Fahrenheit, which is 22 Celsius, and we have blue sky with some white puffy clouds. And the air quality has not been great, but it's better now than it was this morning. It's down to moderate. And they were saying how polluted Montreal was just yesterday and the day before on our newscasts. So, yeah.
It was very polluted. We actually had the air quality score was like in the red. I tried to go for a run and 3 kilometers in, I gave up. So it was pretty bad.
Wow. Also joining us from Madison, New Jersey, Breanne Barker.
Hi, it's great to be here. It is 72 Fahrenheit and very cloudy. We also had the same huge rainstorm yesterday that took it from high 90s down to 70s. And I will take the 70s right now.
Well, maybe they won't take you.
Ugh.
Also joining us from College Station, Texas, Jolene Ramsey.
Hello, everyone. It's a little cooler here in College Station than it is in Austin. I was expecting a high of 101, but it's only 96 degrees Fahrenheit, which is
Wow.
36 Celsius, which is almost the temperature of my incubators, so we could do experiments outside.
Yeah, except that if they're 24 hours, you get a little oscillation, right?
That's true. That's true. Except it's still pretty hot at night.
Yeah.
My
So
first thought was if you're going to do experiments outside, you need CO2, but you don't.
I do
You're
not.
prokaryotic. That's cool.
Yep.
That's right.
Don't need any of that phenol red stuff.
No, you don't need that fancy stuff.
We have 6 people, so we're just missing Alan Dove. And in a way, it's good because we have a nice grid of 3 by 3. I don't know where we'd put the 7th person. By the way, when you have so many, so each of us is individually recorded. So then the video editor takes all of this and, arranges them and does things. And then to output the final product, you have to render basically 6 separate video streams. So it takes 6 times longer than if you just had one.
Wow.
Or 2 or 3. So it's always nice to have 2 or 3 for him. It's much faster. So now it's going to be past midnight on Saturday.
Thank you.
When he gets it done.
Exactly. Thank you.
Well, it depends when he starts, right? He doesn't want to spend his Saturday editing TWiV. I don't blame him. Thank you, David. If you like these programs, we would love to have your support to continue them. We are a nonprofit entity here in the U.S., and we do not do advertising. We take only your donations to make these programs, these science programs. So go over to microbe.tv/contribute. Just 2 news items today. We didn't have any last time. One, I put the PDF in there for everyone, right? Because it's a Wall Street Journal article, which is paywalled. This is entitled, Trump Administration Puts New Chokehold on Billions in Health Research Funding, by White, Subaraman, and Thomas. So the bottom line here is that the NIH will not be able to award grants to outside researchers under this new restriction for the rest of this fiscal year. So, as you know, NIH has research on campus. It's called intramural research, and then it does a lot of funding of external labs, and that's called extramural. So they're saying the extramural funding can't happen. That's like $15 billion. And this is ordered by the director of the Office of Management and Budget, Russell Volk, who was a co-author of Project 2025, which says the NIH is a mess and needs to be restructured, blah, blah, blah. Just ridiculous.
Actually, the subsequent news item that you've picked, if I'm not mistaken, says that within, I can't see the link here, but I'll find it. Says that apparently within about 24 hours, that pause, if you want to call it that, was reversed.
Yes, that is
okay.
Also what I saw on social media.
I think, which is interesting, okay, because I think that, basically a bunch of senators and who knows, congressmen, who knows who else, went berserk, said this is crazy.
So that's just a committee. That's just a Senate committee vote.
At any rate, pressure was put on the White House. There was a blow up, and they reversed that thing.
Yeah.
Okay?
I thought it was within hours, not even 24. I thought it was very fast. But
as I read these, well, let's do the second one, then I have some comment.
Okay, so the second one is, this one is in Nature, which you can see part of it, not all of it. Congress has your back. U.S. senators tell scientists they want to protect the NIH budget. So this is a committee vote, U.S. Senate Appropriations Committee, and they voted to reject Trump's plan to cut the budget by 40%. That's a different issue, right?
Right, yes.
Than this canning of the rest of the fiscal year extramural funding. So this committee voted to not to cut it. They're going to give it a 1% increase, $400 million. And Trump also wants to collapse the 27 institutes to 8, which is also from the Project 2025. But these guys, these committee people said, no, you don't want to do that. But it's got to go to the rest of the Senate. It's got to go to the House. They have to agree. And then it has to go to the president for signature. So I don't know what it's going to look like by then, right? It's nice that this committee wants to do this.
Yeah, and it's a bipartisan committee.
Yes.
Okay.
It's just confusing. It's very confusing. No funding. No, there's going to be funding. No funding. You can't say there's no funding. And then some people have lost their grants and some people are supposed to get their grants back. But how do you do your work when you don't know if you're going to have funding or not, right? Graduate students have to be budgeted for a whole year. Postdocs don't have any safety nets. Supply costs are going up. It's really hard to plan when things are going back and forth, and we don't watch every bit of news, but every, in the classic style, the news is changing every day, multiple times a day.
Yep.
it's really, I'm glad that people are paying attention to it, because the scientists can't be the ones to fix the funding problems and do the science.
Yeah,
what
but
interests me is, I mean, this sort of chaos is, of course, what characterizes our time, because one day there's tariffs, the other day there's not a tariffs. One day we're kissing up to certain politicians, other days we're not. And so it's chaotic. What interests me here is the notion, I mean, it hasn't come to pass yet, that, I mean, people like me are waiting for somebody to stand up for sanity, okay? And it hasn't happened. And yet there is this pushback from this particular committee. It would be fascinating if ultimately the thing that made Congress become responsible was cutting science funding. Wouldn't that be interesting?
Yeah.
It would. I know that there have been a few things that have gone around, and every time I see them, I feel like I wish they were getting a much larger audience, where people are talking about, how much science funding, how much NIH funding, whatever, goes to your congressional district or to your state. How much are these cuts actually impacting your community? And also people thinking about, how many pharmaceutical drugs, how many different things that you encounter every day were funded by all of these things. I think if people understood some of this impact, and I think, how much money my community gets as a result of all of these things, I would hope there would be more outrage. But then again, I can't predict anything lately.
Congress has historically been good to NIH, and the president has signed the appropriations bill. I don't know if we assume that Congress is going to be good again. I don't know what Trump will do, but to override a presidential veto takes more than a majority, and it's very difficult to get. So I don't know what's going to happen.
It would be very interesting to see whether or not Congress, Trump would actually veto something that this Congress passed like this, okay? Because, he's pretty concerned about his standing.
Yeah, we'll see.
Interesting.
Yeah, but he also, comes back at all the people in Congress, and if he doesn't like what they've done, he'll say, well, I'm going to support someone else in the primary.
Right.
In your district or whatever. So,
well, what we need is a critical mass of pushback. I'm always thinking of who was the guy, the McCarthy. The McCarthy era, where he had everybody in the palm of his hand for the longest time, until a couple of people pushed back enough. A couple of respectable people pushed back enough, so that it started a landslide towards the emperor has no clothes, and that did it. But people are scared to do that. They don't want to be the first one out there. And Jolene, I really, I'm sorry, but I really appreciate your perspective.
yeah.
I don't, I only deal with this now in theory. Okay, you deal with it in reality.
Okay.
Yeah, but I think, for academics, it's hard for the professors, but it's harder for the trainees. I think that's the part that really is concerning, because there's hope that things will come to a place where we know what's going on and we at least can predict what will be next. But that doesn't come immediately.
All right, on to the science. So the snippet today is, as Angela said before, OG molecular biology.
No abstract. No abstract even needed.
It's great. It's a 1958 paper. And the reason we are doing this, so this is the famous Meselson-Stahl paper. And the reason we're doing it is because Frank Stahl died earlier this year, died in April, actually, and the obituary was published in July. And I was at a textbook meeting, and my co-author, Ann Skalka, she knew him. She said, I was texting him just up until he died. He's such a cool guy. He was in his 90s, right?
Mm.
So before we get into the science, his background is very interesting, the background of him in the paper. So he was born in Boston. He got a BA at Harvard in 1951. Then he got
Because
a
he
PhD.
didn't have to commute by
Yes.
going to Harvard. Otherwise, he really wanted to go to Brown.
Oh, really?
Yes.
That's probably from the Times, right? I'm reading the
Yes.
Cold Spring Harbor one. Then he went to the University of Rochester to do his PhD. And while he was there, he went to a molecular biology course at Woods Hole being taught by Watson and Crick. Can you imagine?
Amazing.
And so there, Stahl met Matthew Meselson, the other author on the Meselson and Stahl paper. And they, during a break in the course, Stahl was sitting under a tree drinking and selling gin and tonics. And Meselson introduced himself to him. So Meselson was a grad student at Caltech and liked to develop methods of experimentation. And Stahl apparently knew some math. And so they wanted to design some experiments, and they hit it off. And so they said, come to Caltech. So Stahl went to Caltech, where I think they were both postdocs with Linus Pauling. Is that correct?
I think that's correct. I think actually Meselson did his PhD with Pauling and got his PhD a year before this paper was published. And he hung around at Caltech. So one of the things I read, maybe it's the one you're reading from, said that at the time they were both postdocs. But very interesting. Here's two postdocs publishing a PNAS paper, and the PI, Sal Luria, Linus Pauling, is nowhere to be seen on this, which is not, I'd say it's relatively rare, but not unheard of at that time.
Joan Stites did that with Watson, right?
Yeah, she did that with me.
You too. Yeah.
Ed and I have a paper that we published when I was a graduate student and he was a postdoc in different laboratories. I think Joan led that charge and set the example and said, you guys did this. This was your idea. I'm going to stay out of it. And she learned that from Watson,
So this is a really cool story because the two of them they end up figuring out how DNA replicates. They also show, as we'll see, that when you heat DNA, the strands separate, which nobody knew up until that time. And they also developed density gradient centrifugation, which we have all used, maybe not Angela, I don't know.
I have.
You have.
I have.
And they bloody invented it, which was you take cesium chloride solution, which is quite heavy, and you centrifuge it at very high speeds, and then the first ultracentrifuge had already been developed. And as it's centrifuging, it forms a gradient in density. And if you have mixed any kind of macromolecule in with it, the macromolecule will equilibrate at its own density in this gradient. So that's why it's called density gradient centrifugation.
Absolutely brilliant and so much fun. Cesium chloride gradients are so cool, especially if you have DNA that's stained with ethidium bromide.
Right? So then you use it to purify. I used to use it to purify plasmid DNAs for years. You get beautifully pure preps, and you mix it with ethidium bromide, and when you shine a UV light at it, it's red, and you can see it, and you can pull it out of the tube.
I know, but pulling it out was really hard, at least for a young graduate student.
It's a technique. You have to do it over and over again. It's like surgery. You do it 100 times, and you don't kill the patient anymore. Right?
So when you purified plasmid DNA, you had to stain the DNA. You couldn't just see a band.
Correct.
Yes.
Depends. It depends on the copy number, right? But often it didn't grow well because whatever gene you'd put in was toxic, right, to E. coli. So you had to shine we had these handheld UV lights, and you would shine it on the side with one hand, and then the other hand you'd poke a needle on just under the band and pull it out.
We had our UV light in a ring stand, so we had two hands. One hand to go this way so that you weren't poking your fingers.
We did that too.
The next cool step for me was always doing the dialysis, where you dialyze the cesium chloride out, and you could just watch the cesium chloride pouring out, the Schlieren coming out of your dialysis bag. I would always drag people in. Come watch this. You can just see it streaming out.
See, and this is if you have made sure that when you use your needle, you did not somehow inadvertently end up spraying ethidium bromide containing solution all over the lab.
Yeah, it was a skill. There's no question about it. And I did that for years after these quick prep kits were available because it was just so clean and it would last forever. It would just last for 20 years. It wouldn't degrade, and it was beautiful.
When I closed down my lab a year ago, I still had the saturated stuff to do all the extraction and dialysis and all that stuff. Yeah. Best
So
DNA.
anyway, at the time, in the 1950s, as everyone may know, in 1953, the structure of DNA was solved by Watson and Crick, and they said that DNA has two strands, and they're joined together by specific interactions between the bases, of which there are four, and there are two pairs of bases. And so they said, A pairs with T and G pairs with C, and it makes this long double helix. And at the end of their paper, their Nature paper, they wrote, "It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material." So basically, this double strand could come apart and the strands could be copied. But what we didn't know was, were both strands copied? Was one strand copied, or was there a patchwork? So there are three ideas for how the DNA was copied. There's the semi-conservative model, which says the two strands are taken apart, each is then copied, and so now after one duplication, you have both strands have been duplicated. And then there's the conservative model, where after the first cycle, each is one of the strands is copied and the other one remains duplex. And then there's this dispersive model where there are patches of copying. Not each strand is not copied in its entirety, but it's copied in patches. And so Meselson and Stahl, with their density gradient centrifugation, which can figure out can see differences, small molecular weight differences among molecules, they said we can solve this problem, and that's what this paper is all about.
And if any of our listeners need to see this visually, they can look in any molecular biology textbook where this experiment is always described. And there's sort of a classic figure that most of us think about, where you can see, the semi-conservative model where the products contain one old strand and one new strand. The conservative model where the old strand stays together and you make a completely new strand, as opposed to semi-conservative where it was sort of like half and half, and the dispersive where it's sort of scattered fractions.
So the paper is published in PNAS, Proceedings of the National Academy of Sciences. It's called The Replication of DNA in Escherichia coli. Meselson and Stahl from Caltech. And it's interesting that there's no abstract. The first sentence, they spell bacteriophage instead of bacterios.
Interesting.
It's interesting, right? I guess they changed that later, right, Jolene?
I think there's variations in the literature, and I always assumed that was partly based on like who people were taught by or what their English origin is.
I want to note as well. This is a paper that's Proceedings of the National Academy of Sciences. So the way these papers are done, it's not it's a little more complicated nowadays, but especially back in 1958, there are members of the National Academy, and they can communicate papers that are not their own that get published. So they can say, "Oh, this looks cool. I want to make sure these guys get this published." And there's a review process and stuff, but that person champions it. And the communicator of this paper is Max Delbruck, who is sort of the great grandpa of phage biology, really, and molecular biology, okay? He is absolutely a historic individual.
So they start out by telling us that they want to figure out the mechanism of DNA replication using this technique they developed, density gradient centrifugation, which can pick up small density differences among different molecules. And the way it works, there was an ultracentrifuge available at the time, which was called a Model E. It was the size of 2 refrigerators. I saw one.
I'd say
I
3.
actually used one once.
I would say 3.
3 refrigerators.
Yeah.
It was very long, yes.
Sounds terrifying.
And
Yeah.
there was just one place where you interacted. It had a very special rotor that it would spin fast. You would put it in the machine and it would spin. But the rotor didn't take tubes. So today, most ultracentrifuges have a rotor which you put tubes in, and you could put cesium in and bring it to equilibrium or whatever. But this had a little cell made of quartz that you would load the sample in, put it in the rotor, and then put it in the machine. And then as it's spinning very fast, you could look through an eyepiece, and you could look into this this quartz cell, right? It's going so fast that you don't notice that for a fraction of the cycle, the quartz is not in front of the lens, right? And so you can actually watch molecules moving down the gradient by their Schlieren pattern, as Cassie was saying before, with the cesium chloride coming out into the water, right? It's more dense. It makes this Schlieren pattern, which you've seen in other, if you put one kind of liquid, if you put a syrup in a drink, that's the Schlieren pattern, right?
I put a link in the show notes here that describes the machine and also has a picture of the rotor and describes the optics. And in this particular case, in lots of cases, you can actually shine lights of a specific wavelength on the cell. And in this case, since nucleic acids absorb UV, they're shining UV on the cell, and then it goes through and goes to a detector. So basically, I guess the band that you see is a shadow from the cell where the DNA is sucking up the UV that's going through the cell. And for I don't know if this is for the lay people or for all of you others. I don't know if you've ever thought about this, because I learned this in my physical biochemistry class in graduate school. If you look at this diagram, it shows you how Schlieren optics gives you peaks, okay? Because if you're doing a sedimentation experiment, you get a gradient of macromolecule concentration across the cell, which would just look like a kind of a gradient. And what the Schlieren optics does is it's an optical way of taking the first derivative of those curves. So where they are steepest, you get a peak. And where they're plateaued, it's level. And it shows that in this. Some guy demonstrated this in this biophysics class. I'll never forget it.
You also put a link to a nice article by Elio Schechter.
Right. That's the one that has the
somebody did.
picture of the Model E. Yeah.
And that's a great article because
Yeah.
in
Oh,
his
yeah, yeah,
usual
that's the
way, he says, in the old days, it was hard to do experiments.
Yeah.
Can't
So
just do RT-PCR.
TWiV listeners will recognize Elio Schechter's name. So.
Anyway, in this paper, the very first figure is just a Model E experiment, a banding of E. coli DNA with time, with hours of centrifugation. And you can see that initially the DNA is dispersed throughout the cell, right? Because you mix the DNA with the cesium, you put it in the cell, and you start the rotor. And then once it gets up to speed, the gradient begins to form. And they have they have pictures taken at different times of the cell showing how the DNA is coming closer and closer and closer and forming a band at its native density, right? At the very last time point, 43.5 hours, you can see it's a rather wide band, but the DNA has come to its own density in the cesium chloride.
I want to jump in here and say the same thing happens when we made our plasmid preps. Everything would be completely homogeneous at the start, and then you would centrifuge your stuff and it would band. But at some point we started using what were called vertical rotors, and so stuff was banding across the tube in a different way. And so that was really cool. And also, for those of you who may be impatient and made it through the weather and now made it this far and are thinking, are they ever going to talk about the experiments? But this paper was first quoted, the biochemist John Cairns, a director at Cold Spring Harbor Lab, later head of Mill Hill at ICRF in England, was the first to call the Meselson-Stahl research, quote, the most beautiful experiment in biology. So this is kind of why we're spending a lot of time on this, because it's just so fascinatingly beautiful and simple and elegant.
We were also talking a couple of episodes back about doing classic papers.
Mm-hmm.
And this is one of the classic papers. It's so much fun.
It's the first time I had ever read this paper was yesterday.
Yeah, actually, I hadn't read
I'm
it
not going to
before.
lie.
It's good.
I'd never read it.
It's not a hard paper to read, right?
No.
It's-
No, and I understood it, and I'm not a specialist by any means. And I think, explaining it to the listeners how, like Kathy said, how beautiful and simple it is, but at the same time, the idea to do it maybe isn't as simple, but I think what they did was actually quite beautiful. It's true.
So back to the problem about how DNA reproduces, right? We had the 3 different ways, and they said, well, if we could make one strand of the DNA heavier, we could see the strands separating on this, or the DNA separating differentially on this method that we have just made. So they decided to use heavy nitrogen. So normal nitrogen that's in the air is isotope number 14, and 15 is the heavy version, which they thought, well, this would make the DNA heavier, and therefore we could see a difference. And so they actually did that experiment initially. You just take E. coli and you add either nitrogen 14 or 15, so they had been able to buy a salt with the nitrogen of either one, and then they run it on their Model E density gradient, and this is figure 2. You could see N14 labeled and N15 labeled DNA get beautifully separated. 2 peaks. It's a beautiful, simple experiment. I wonder how many times they did it to get this beautiful. Right?
Actually, in the methods, I got the impression they didn't do it that many times.
Yeah, it probably worked easily. Yeah. So there's a small difference. You can also do a tracing of the two bands, and you can get 2 peaks which are nicely separated. So it can work. N14 and N14 and N15 can be distinguished in DNA.
And just a technical note, because I thought this was interesting. They don't purify the DNA.
No, they don't.
They just harvest the bugs,
right,
and add a little detergent to lyse them and mix them with the cesium. You do the same thing essentially with plasmids, right? With some of the preps. There was a few more pre-steps. But at any rate, really easy. Just lyse it and mix it with the cesium and run the equilibrium gradient.
So the real experiment they take bacteria and they add the N15 compound and they grow them 14 generations in the presence of N15. And then they switch to N14 containing medium. They give a 10-fold excess over the N15, because now the idea is that the DNA all has N15 in it. And so now they added 14. What's going to happen as the DNA reproduces?
Yeah, so they have the old DNA marked as heavy, and potentially any new DNA that's made will be light.
Right. So then they lyse these, they take samples from the culture and they lyse them and add them to to their ultracentrifuge. And that is, then they photograph, they do UV photographs during the course of each centrifugation, and they can also scan them so you can see peaks. But the photographs that they take are just beautiful. So you can see the figure 4 is the bloody experiment, time 0, where you have all N15 DNA. The DNA is one band at a certain place. And then as you go to 0.3, 0.7, and one generation now, it has completely shifted to a different place.
So what we mean by generation is the amount of time it takes to replicate the DNA completely.
Which,
and,
isn't it like 20 minutes, Kathy?
It depends on the exact conditions, the media and everything. And I don't even remember if they stated what their generation time was.
They have a growth curve here.
Yeah, they do. They say that it's 0.81 hours in one experiment and 0.85 hours on the other.
Right.
That's pretty long.
So longer than I thought.
Yeah, I mean, they're growing it only with, like, they're growing it in a weird media because of
a minimal
nitrogen.
medium,
You said 0.8 hours?
0.8 hours, yes.
Yeah, that's not that long. That's like 40 minutes. That's not that
Okay.
unusual for an E. coli generation time. You can make them grow faster, but actually, if they were growing faster, then they'd have to be replicating more than one copy of the DNA at the same time. So that would complicate the interpretation.
Interesting.
So that's a good point, Jolene. So the chromosome of E. coli, the DNA is a single molecule, right?
Yeah, circle.
A single circle. So what we're looking at here is this circle being copied. So this figure basically solves the problem. They say, so until one generation time has passed, you get half labeled molecules. And the fully labeled DNA is depleted. One generation time after the addition of N14, these half-labeled or hybrid molecules alone are observed. That's the shift. And then subsequently, only half-labeled DNA and completely unlabeled DNA are found. Because remember, beyond one generation now, you've only got N14 in there, and so you're getting unlabeled DNA.
That experiment there distinguishes between the three hypotheses.
That's right.
Because
Yeah.
if it was the conservative mechanism where you keep the parental thing and make new progeny, you'd have two bands after the first,
Right.
still the original labeled guy and a completely light guy. If it was the dispersive mechanism, you'd see the same thing after one generation, but after two generations, you would see all hybrid molecules, and yet they see some hybrid molecule and some that's not labeled at all, okay? So that rules out the dispersive. And they say in the paper, I love this, in their discussion, that this rules out Delbruck's model, okay? Now, I don't know which that was, but it was probably the dispersive one. I'm not
it
sure.
was. I looked it up because I
Okay,
was
good.
curious about that.
And,
And yet he
He's exactly the one
communicated
who
the paper.
communicated it.
This is science, right? Oh, you proved me wrong. Let's publish that. Yeah, it's great.
It's the way it should be. Yeah.
At least he was a good sport about it. Others
So-
might not be.
So they're just there are four figures, and then they go right into the discussion, and they say one sentence: these results permit the following conclusions to be drawn. No drawn out introduction to the discussion where they repeat everything. So first, the nitrogen of a DNA molecule is divided equally between the two subunits, which remain intact through many generations. Right? Two, following replication, each daughter molecule has received one parental subunit, defining that all DNA molecules are half-labeled one generation after the addition of N14 shows that each daughter receives one parental subunit. Number three, the replicative act results in a molecular doubling. And they have some nice pictures where they first illustrate this with little rectangles, and then they use the Watson-Crick structure. And they say these results are in exact accord with the expectations of the Watson-Crick model for DNA duplication. And they show the double helix in terms of their heavy and light nitrogen here.
The two model versions of this that they show here, I've definitely seen before.
The other,
Yes, these have been copied by other people. Yeah, for sure.
The other interesting thing is that I had forgotten about this, and I used to have to point it out to students that in older papers, particularly in science, the reference number would sometimes lead you to what was really more of a footnote. So that conclusion on number 2 about the finding DNA molecules are half-labeled one generation time shows each daughter molecule receives one parental subunit, and that's footnote 14, and where it also says, this result also shows that the generation time is very nearly the same for all DNA molecules in the population. This raises the question of whether in any one nucleus all DNA molecules are controlled by the same clock, and if so, whether this clock regulates nuclear and cellular division as well. So it's like there's this extra stuff in the footnotes, to have you think about, and you might miss it.
And also, one of those things that, for various reasons, they had to do it this way, but a nice part that comes out of doing this in E. coli, where this might have looked a little messier in other cell types.
Yeah.
So Frank Stahl went on to a career in what was then the new Institute of Molecular Biology at the University of Oregon in Eugene, and spent his career there. And Matt Meselson, who is still alive, and is 95 years old. Went to Harvard.
Wow.
Mm-hmm. Right. But the other thing they did in this paper is not ever mentioned, but really important. So people have been experimenting with DNA, and one of the substrates was E. coli DNA, easy to get, but they also would use salmon sperm DNA. I mean, I remember going to the lab once, and we have these bottles of salmon sperm DNA. It's like, why do we need? Because you can get a lot of it, right? And it previously been shown that when you heat this, something happens to the DNA in E. coli or salmon sperm, and they differ a little bit. But they say heating brings about an abrupt collapse of the relatively rigid and extended DNA molecule. And people were trying to figure out what was going on. So they simply took their labeled DNA and heated it and ran it on this gradient. And they show that basically the two strands are separated by the heat. It's denatured.
Mm-hmm.
And you can do that beautifully with N14 or N15 labeled DNA, right? Or hybrid DNA. And they do
The
that.
hybrid DNA is the critical one because you heat that and run it and you get two bands.
You get two strands.
One's heavy and one's light.
Mm-hmm.
So this tells you heating just separates the two strands. And the cool part is if you lower the heat, they come back together again and they renature. And those that was studied extensively. One of the professors in grad school when I was there, Jim Wentmer, had worked with people who figured out the kinetics of renaturation, and you could figure out all sorts of things about the DNA. Remember, before you're doing any sequencing, you could find.
So they know the difference between coli DNA and salmon sperm DNA in that regard. And is that because the salmon sperm DNA is complex, sufficiently complex, so it's not renaturing? Is that the idea?
I read something about that because I was confused why they were making this point. And apparently the sensitivity of their assay was just such that they couldn't detect the salmon sperm DNA coming apart. But it does denature just the same.
Sure.
Yeah,
Okay.
it's definitely longer strand, like pieces that you're getting with salmon sperm.
Yeah, and of course it's longer, different sized chromosomes and so forth. Yeah. But they all denature. DNA denatures all the time, and the kinetics depend on the percent base content and so forth. So this is the paper, and many classes used to teach this routinely, right? Because it's just such a beautiful and simple experiment. I don't know. Brianne, do you teach this experiment anymore?
I don't teach a class that would do this. That's a prereq for a class I teach, but they do talk about this. And I remember drawing these figures in my notebook when I was taking the classes.
So here's a question for all of you. Today we have, in 2025, with all the technology we have available to us, let's say that this hadn't been figured out yet. How would you do it?
I don't know, like somebody, Angela was saying, the insight and the creativity to come up with the experiment itself. Once you know it, okay, it's obvious. But if you didn't know it, to come up with this as a technique, I don't know how that happens. I remember trying to figure out how DNA was going to be sequenced. All right. And it turns out to be beautifully simple. But we spent, months trying to figure out how you would do this, and we didn't have a clue. So trying to think another technique for doing the same thing. I don't know.
Yeah, the only thing I can think of is sort of fluorescent labeling.
Mm-hmm.
But I would say that that answer only comes from knowing this work. I don't know if I would have thought of fluorescent labeling without knowing this work.
Yeah, well, I thought of radioactive labeling for the same, but with the same caveat.
Yeah, you would have to. You have to label the strands, right? Differentiate. That's the whole key. And N15 is beautiful because it's an atom, and it's not going to mess up the DNA structure all that much. You can imagine that you could take a base with some kind of modification that was fluorescent or some biotin, but you could get arguments that it would mess up the DNA. So I'm not sure it would be terribly different from this experiment.
And it's true. You read it and you're like, oh, it seems obvious. But imagine having to think about doing that and like putting N15. Also, I don't know how readily available it was. I mean, I'm not aware of how available it would have been then, or how stable or in their media, or maybe they have to optimize the media conditions, because for the E. coli, like, I have no idea.
I'm not sure I know how available it is now.
Well, probably not that available. I don't know.
It's not hard to get.
In general, back then, it might not have been so easy to get these things, right?
So you were asking about teaching it, because I was thinking about this as I was reading this. First of all, I sent the paper to my buddy Ed, okay, because, you know, he and I kind of grew up together, and I thought he might like to look at this again. But one of the reasons I sent it to him is that I very distinctly remember the day that my DNA replication lectures had become sufficiently crowded so that I decided that I wasn't going to teach the Meselson-Stahl experiment. Okay. And I remember telling Ed that, and us kind of standing around moping at each other and mourning this particular landmark.
You know, the other thing I'm noticing here is there's no methods section.
I was also looking at that, or supplementary.
It's just incorporated into the entire paper.
Yeah, isn't it usually sort of in the figure legends?
Those were old Nature papers.
Yeah.
They were all in the figure legend.
But here, they're also in the discussion as well, yeah. Oh, the supplemental, that's not very old.
No, that's pretty recent.
That started when I was a grad student.
Really?
Oh,
wow.
Okay. There is a range in our ages here, I think.
When did this footnote, so here from what I can see, it's either a footnote or a reference, and there's no way to know until you actually go to the references. So when did that change, and how was that ever?
Not
Actually,
soon enough.
they did the same thing. I remember seeing the same thing in both Science and Nature.
Right.
And that went on for quite a while. I don't know that it does still.
I would say maybe in the 80s.
Yeah, I was going to say, I think I've read 70s papers that still do it this way.
I still have seen papers from, I would say, at least in the 90s, where you'll have a reference number at the end of a sentence, and you go to the number in the references cited, and it says personal communication so-and-so, or so-and-so unpublished, or in preparation. And I'm like, no, I'm stuck.
Right. But the kinds of footnotes that had additional information in them, content-wise, like this, maybe it ended before the 80s, but I definitely remember teaching students that they had to look at papers and look at the reference sections.
I have another. I'm sorry.
I was going to say, there's a paper that I teach that has the methods in the figure legends, and that paper is from the early 90s.
Yeah.
Oh, wow.
I have another little story / semi-boast here, and that is that I met Max Delbruck. It must have been somewhere between 1970 and 1975, because I was a graduate student, and believe it or not, I was working on Phage, and Yale was right across the Sound from Cold Spring Harbor, and so I used to go to the Phage meetings, and talk about my work. So the Phage meetings were kind of old hat at that point; they weren't the original stuff. But I went to one of these, and I was sitting out on the lawn at lunch or something like that, and lo and behold, sitting next to me was this guy, Max Delbruck. Okay, and he says, "So, Max Delbruck, by the way, he and Luria kind of started molecular biology by studies on phage, and they were the ones who isolated and identified what we know as the T phage, T1 through T7, I guess. I don't think there's a T8. No. Okay, so and Max says to me, so." What phage do you work on? And I said, T7. And he said, is that the one that makes its own polymerase? I said, yeah, Max.
Cool.
Awesome. Just awesome.
So one more thing about this paper, the funding, NIH, okay, this is 1958, and the National Foundation for Infantile Paralysis.
Interesting.
Which was originally set up by FDR to make polio vaccines, right? But at the end of the 50s, they were trying to broaden their support, and they support fundamental science like this. It's great.
March of Dimes, right?
I was wondering.
Yeah, the March of Dimes was the funding mechanism for NFIP, right? Which still exists. It's now the March of Dimes.
You could actually, and I remember doing this, you could buy a little cardboard thing with slots in it and stick dimes in it.
That's right.
Okay. And collect a bunch of dimes till you had a certain donation and then send it to the March of Dimes. I've done it.
Wow.
When I first started my lab, I looked into getting some polio support, but they had just stopped supporting polio research, and they were moving into other things. Would have been cool. All right. Well, we're going to continue talking about DNA, aren't we, Jolene?
That's right. Yeah, that was a lot of fun. And, viruses have DNA and they replicate the same way that cells do. And we're going to talk about a paper now. This is, of course, a phage paper. And this paper is published in Cell Host Microbe this year, 2025. It is called Sequential Membrane and Protein-Bound Organelles Compartmentalized Genomes During Phage Infection. And it comes from a few groups, one primary group based at University of California, San Diego. There were two co-first authors, a grad student and a postdoc, Emily Armbruster and Pulwanti Rani. And there are a lot of people that contributed in between. And it comes jointly from the labs of Elizabeth Villa and Joe Poliano. And there are several other PIs and others who contributed from all over the place, really. And so in this paper, they're studying jumbo phages, which are phage with large genomes. Usually the cutoff is around 200 kb or bigger. And these are phage that are known now to have this large protein compartment inside the cell where the phage DNA is sequestered away from the rest of the bacterial cell. So that's one really cool thing we already knew about some jumbo phages, especially ones that infect Pseudomonas. But in this paper, they're looking at another compartment inside of a bacterial cell that's a lipid-based compartment that holds the DNA. And that's
not supposed to be in bacterial cells. No.
I also decided to call this a phage fortress. I thought that was cool. Not the vesicle, but the actual nucleus itself, the sheath that it creates. Also, I know so little about phages, and after reading this paper, Jolene, I'm now a fan of phages because this is really cool.
I'd like to jump in and say that for those of you that might not think about bacteria that much, we don't generally think about them having compartments. That's one of the things that distinguishes eukaryotes from prokaryotes, and bacteria being prokaryotes, is that eukaryotes have a nucleus, which is a compartment, which is where we generally think of DNA replication occurring, and bacteria don't have that. And so the fact that they have these jumbo phages have this thing that is now called the phage nucleus is a linguistic thing that is allowable because bacteria themselves don't have a nucleus. And so you can talk about this other nucleus and co-opt that word. And so finding this other compartment that Jolene has just alluded to further complicates or makes this interesting.
Yeah, I mentioned there was a talk about some similar types of things at ASV, which is where I learned a bunch of things about this. And the protein-bound phage nucleus, I'm sort of cool with. In thinking about things I teach in microbiology, because I can think about other compartments, and I always say, yes, oh my gosh, guys, there are compartments, but not lipid compartments. Mm-hmm. And now the thing that really sort of shocks me here is the fact that it's actually a membrane-bound compartment, which seems to be, really crossing that line.
Yeah, so it's not unheard of, like you said, for bacteria to have compartments, especially protein-bound compartments. Things like the really cool ones I think of are magnetosomes. There are also some lipid rearrangements that happen inside of bacteria to help with things like photosynthesis. But the real big novelty here is that there's DNA inside the compartment on purpose. The phage is doing it, and it's not just hidden in there. It's actually actively being transcribed. And that's the big interesting thing here.
So it's kind of like a eukaryotic nucleus But inside of a bacterium that was built by a virus.
Correct. Yes.
Which is amazing.
So, Jolene, I have a peripheral question. In their cartoons, they picture these as tailed phages. Are they tailed? They are.
They are, yes.
They must be huge.
Yeah.
In a relative sense.
Yeah. They are big. They have big heads for sure because they have large genomes. And
So
it's
I
a
guess-
myophage.
How big is T4?
T4 is about 160 kb.
Okay, so
And this
relative, it's
one,
not that
the one
much
in
bigger.
this paper is called Gosler. It infects E. coli, and it has a 237 kb
Okay,
genome.
so bigger but not crazy.
Jillian,
Yeah.
where did the name come from, Gosler?
I do not know the answer to that question. I have a feeling it has something to do with where it was isolated from. It comes from a duck fecal material, and it infects a chicken peritoneal isolate. I do not. That's my
Gosling?
guess. I don't know.
Maybe it's somebody's cat. Who knows?
Possible. Okay, so in this paper, they're studying E. coli phage Gosler. They're looking at what is happening to the DNA once it gets inside the cell. There were a couple of other studies in Pseudomonas, another kind of gram-negative bacteria, and jumbophages that looked like they had these lipid vesicles. It wasn't clear if the lipid vesicles inside of the cell had a role in the phage infectious cycle or if it was a host defense that was allowing the phage to continue its, preventing the phage from doing what it normally does. So what they did was they started out calling these vesicles that had previously been observed the USB, which I've now forgotten what that stands for, the unidentified
spherical
Body.
That's
Thank
it.
you. Yeah, so they know that the phage delivers DNA into the cell, and they also knew that although these are DNA phages, these jumbophages are resistant to all host defenses that target DNA. So CRISPR was the big one that people had been looking at, but also restriction modification systems are not active against these. Even though the phage is putting DNA presumably into the cytoplasm, these weren't working. And so that was consistent with this idea that the phage was somehow protecting its DNA before it built that phage protein nucleus, because that takes some time.
By the way, Goslar is a city in Germany where it was isolated from duck feces.
Oh,
Okay.
it's the city.
There you go.
Okay. Thank you.
See, my silly comments about Saved by the Bell, we're just not going to be working here.
So the phage DNA is delivered, and just as a kind of overview of the infectious cycle, is known the DNA got into the cell. Eventually, the protein nucleus was built, and the DNA gets inside that protein nucleus. More transcription happens. These jumbophages also make these tubulin-like proteins, and in Gosler, these tubulin filaments will all converge from the periphery of the cell onto the phage nucleus and then make it rotate. And that is thought to be important for its ability to fill phage capsids with copied DNA. And then eventually those newly filled capsids will get their tails and the cell will lyse open.
Boy, that's weird.
It is.
The
It
rotation
is quite
bit?
weird.
That's very
Insane.
weird.
So the protein that forms this phage nucleus is called chimolin A or ChimA. And that's important for a lot of the experiments that they do. If you prevent the formation of this protein, then you won't get a nucleus and the phage can't make new virions and it can't lyse and get out and do everything it's supposed to.
And that's encoded in the phage genome, right?
Correct. That's correct. Okay, so the first thing they did was they looked, does Gosler make these unidentified spherical bodies? Yes, it does. Shortly after infection, they could detect using cryo-electron tomography these vesicles that looked like they had some density like DNA inside them. And they then also used a fluorescence labeling technique that goes throughout the paper where they can label different membranes in certain colors, and then they label DNA. And so they label the full outer membrane, so basically the periphery of the cell, so we can see in the images that they present in the paper the outline of the cell. They label all membranes, and this is important because then if a little vesicle is formed after the phage infects, then they can see that as a dot in their images, and then all the DNA. So that means the host cell DNA, which is filling up most of the cell, and then any phage DNA that gets delivered, they could see as well. And so they show that within 10 minutes after infection, they see a lipid dot and they see a DNA dot, and they are in the same place in the cell. And they see that those stay together as the infection progresses, and that membrane signal moves next to what becomes the phage nucleus, where you see a big concentrated blob of DNA in a part of the cell that becomes slightly wider than the rest of it.
I also really like that we don't see other little vesicles.
Mm-hmm.
I felt a little
it's very
better.
Clean.
Yeah, it's so clean.
Sometimes they see multiple, and that's because the cells are being multiply infected based on how many phage they're adding to the experiment. And then the key that allowed them to do these experiments here is this new CRISPR application called CRISPR-I-ART, which is using an RNA targeting CRISPR complex to bind to RNAs in the cell. And that CRISPR complex is inactive for cleavage, but still binds in a targeted fashion. And so because these jumbo phages were basically immune to all DNA targeting CRISPR, they couldn't do anything in terms of like modifying, but they can bind to the RNA that gets made. And then when RNA is bound by this CRISPR-Cas13 complex, then the ribosome can't bind to the RNA, so protein doesn't get made. So this is in essence a translational repression, or what we might call a knockdown. So they show that when they target this CRISPR-I-ART against the ChimA protein, that they are going to get less ChimA protein. They check this by Western. And they also show that when they knock down the ChimA protein, they cannot detect plaques for Gosler on a plate. And what I think is even cooler that they did is that they changed the sequence of the RNA so that the CRISPR complex couldn't target it anymore. And then they do get plaques made again. So that's a really nice way to overcome their block in translation.
So the complex doesn't prevent translation in that modified one.
Correct. Yeah, it will block translation of the one that's made off of the phage genome, but the one that they add that's recoded, which we call ChimA star, that one can still get translated.
That was that added like in trans with a plasmid or something like that? That was my impression. Okay.
Yeah.
So that also means to me that the inhibition here is specific to that one gene product.
Mm-hmm.
Okay. If you just supply that in trans, everything's okay.
Yep. Right.
So I want to digress just a little bit because they used a term in here that I hadn't heard used in this way before. And it's with this first figure. I can't remember whether this is open access or not. But anyway, it's the figure 1B. So they show what looks like a basic microscopic, it says tomogram, and it's black and white. And then below that is something that they call segmentation. Mm-hmm. And my way of explaining it to myself was that it's like they're kind of false colorizing the various things that they see in the micrograph, but they don't really describe how they do that, except I kind of dug as deep as I could, and it just looked like hand waving some software helped them do it. Is there any way to explain a little better how that works, or is that good enough?
So I don't do ET in general, but my understanding is you have a cell and you take what we call slices. You take images at different focal planes and you have this set of images. And then what they do is they define what each item or object is in each image. And then they go to the next image and they define the object. They go to the next image and define it. And so after doing that for all the images in the stack, then they can go back through and make a 3D reconstruction, essentially, of all those segments, and those will be colored based on how they define them, and that's what I'm understanding we're seeing there with the colors, where they've got the ribosomes, they've got the lipid, and they've got the DNA, and then they've got the cell membranes.
Okay. Thanks.
There's some subjectivity there, though, it seems to me, because
You define the
saying
objects.
you define the object.
Mm-hmm.
Yeah.
It's okay with me. I trust him.
Okay. So the next thing that they do is they characterize this ChimA knockdown a little bit more. Now, we would expect that if ChimA is knocked down, they shouldn't form a phage nucleus, and that is what they see. They do not have the formation of that clear blob of DNA in the center when ChimA is knocked down. And they also show that when they tag with GFP, a protein that they know comes from the phage head into the cell with the DNA, that is co-localized with the phage DNA dot. And they show that when they knock down the ChimA protein, that they're still getting this migration of the lipid, but they're not getting the phage DNA coalescing. And so because of that, they still see lots of host DNA throughout the cell. As far as we can tell, right? It's all DNA. That's one thing that they don't show specifically by sequence that it's phage DNA versus host DNA, but the patterns are pretty consistent. What they do show is that when they measure the amount of signal for DNA in the position where the phage DNA is present, the amount of signal is the same for a phage that didn't get into the cell, for a phage that did get into the cell but is very shortly afterwards, that the same amount of DNA after it gets in. And then the ChimA knockdown, the amount of DNA signal doesn't change for an hour, whereas if they don't knock down the protein, the amount gets there is more. There is more. It occupies a larger space, and there's more signal. And that corresponds, we think, to a replication in the phage nucleus. And then they also show that their ChimA star bypass, the recoded version, they then can get the formation of the phage nucleus larger dot, but they don't quite get as much of the host DNA disappearing. So it's a pretty good bypass, but maybe not perfect. Then what they start to look at is how much of the infected cell has this vesicle. So they rename it from the unidentified USB. I sort of think of it as the UFO. They rename it from a UFO to the EPI, or the early early phage
Unidentified body.
the EPI, which stands for
Early phage infection.
Yeah,
vesicle.
that's what I thought. Okay, early phage infection vesicle. So the EPI vesicle. So they do the cryo-ET technique again on cells that are multiply infected, and they show that the vesicles are there. Sometimes there's more than one. In the ChimA knockdown, the vesicle size does not change over time. The size that it is at 1 minute in a wild-type infection is the same size 90 minutes after infection in a ChimA knockdown, when usually the DNA would have been transferred. They also noticed when they were looking at all these different images that there were ribosomes, a lot of ribosomes kind of in an ordered array next to the vesicles. Now, there are tons of ribosomes in the cell. If you take a look at these images, you'll see that they're everywhere. There are lots of them. That's normal. But these ones are arranged specifically such that it looks like what we would call a polysome, or multiple ribosomes attached to and translating off of one mRNA. And they see that repeatedly next to the EPI vesicle. So they think that's pretty curious, and they later start talking about how they think that that's because inside the vesicle maybe transcription is happening, and so mRNAs are coming out. We do know that proteins have to be made. ChimA protein would have to be made to make the phage nucleus, so this is consistent. they look more closely at those vesicles and they see that it has a bilayer, or that it has the staining that you usually see for a membrane bilayer, such as that of the bacterial membrane that we think it would probably be coming from. And then they label the virion RNA polymerase. Many phage will deliver a virion RNA polymerase into the cell with the DNA and use that in the initial stages of the infection to transcribe RNA from the DNA. And they show that when they label this virion RNA polymerase that comes from the head, it's also in the same position that we see the lipid vesicle as it's migrating through the cell. Okay. Then, so we've got this vesicle. It looks like the DNA is getting into the vesicle when it first gets delivered into the cell, and it moves towards where the nucleus eventually gets. And somehow the DNA that's in this vesicle has to make RNA and make protein so that the nucleus could be made. So they wanted to see if the DNA was transcriptionally active. And our assumption would be that it's transcriptionally active inside the vesicle. So they do.
Jolene, just to give you a break for a second, and also just a little question. So basically what they're hypothesizing is that there's transcription happening inside of at this point, inside of the epi. We're calling it epi at this point. Yes, the vesicle. And that those proteins are then being, like, via the ribosomes that they showed in one of the figures, being made into proteins, that is slowly building our fortress, right? Our phage fortress.
Yes.
Okay.
That's right. Yeah.
Rube Goldberg couldn't have done a better job on this. This is good.
It's so cool.
Yeah.
Like, just for like the listeners to understand, it's crazy that this tiny little vesicle is somehow making its own little protein so it can make a fortress for itself and then slip in without being seen by the cell, basically.
You know, you don't have to make this stuff up. It's wild just on its own.
Yeah.
So I didn't
cool.
emphasize this before, but part of what they were looking at with the size of the vesicle is that it's enough for the DNA to fit, but it's a little bigger than a phage head. So there's room for some other processes to occur inside. And they check for phage RNA. You would expect phage RNA in a wild type infection. Over the first hour, they see the typical temporal program of transcription that you see for phage, where some genes come on right away. We call those the early genes. And then later, a few more genes come on. And then even later, the rest of the genes that weren't on before come on. So we've got the early or middle and late genes that are being transcribed. And then they look at the ChimA knockdown. And what they see is that when the phage nucleus isn't being made, phage genes do get transcribed. There is RNA in the cell, but the pattern of what gets transcribed is similar to the early stages of wild-type infection only. So not all of the phage genes are coming on. About 175, they say, are coming on, which is a lot. So that's a large percentage of the phage genes are being transcribed. They also talk about specifically which genes those are, and you would expect that the ones that are coming on are the ones that would be necessary to make the phage nucleus and start copying the DNA and so forth, but not the ones that are like really late. And late genes are usually the structural proteins that build new virions or new phage particles. Then they see that the ones that aren't changing, the ones that are still being transcribed when they've knocked down the ability to form the phage nucleus, are things like ChimA, expected, as well as some other proteins like PICA, which is a protein that is involved in moving things into the phage nucleus. And they also see that the ones that are not on high levels, or that are not really being transcribed, are those late genes, like the ones that build new virions.
Forgive me for ignorance here. In phage biology, is the description of early versus late sort of the same as you might think about in animal viruses, in that that's dependent on whether it's transcribed before or after replication of the DNA?
I would say that I personally don't make that distinction, but the answer is yes. If you're not copying your DNA, you're not making your late genes, because the late genes are usually made by another polymerase, or they require a middle gene to get made. So yeah.
In T4, late gene expression actually requires DNA replication. That has to do with opening promoters and that kind of stuff. That's
Isn't
part
that
of
true
what's going on.
for lambda also?
I do not know.
Yeah. I mean, I think of your basic card-carrying DNA virus, be it a phage or an animal virus. The onset of DNA replication is going to delineate when late things start getting made, and those are usually what you need to make the virus, capsid, etc.
And there are other things involved. There can be more than two classes, and there are these phage that do two-step injections too, right? And that can influence the gene expression pattern.
That's right.
So it's not always just DNA replication, but
yeah, good question. I hadn't thought about it that way, but I think that's because I don't think a lot about DNA replication, actually, despite how beautiful it is.
well, you're thinking a lot about DNA replication today.
Yes, that's right. Okay, so that was RNA. So they looked at RNA levels when the phage can't make the nucleus. And then they checked just that the protein levels correlate with the RNA, because just because you make an RNA doesn't mean that you're going to make it into protein and so forth. So they did see that when they knocked down the ChimA protein like they had seen before, not so much. When they knocked down ChimA gene expression, not so much ChimA protein. The PIC-A protein, which doesn't have changed transcription, similar amounts of protein, a little bit variable, but similar amounts. And then the other one that they looked at was called REC-A. So REC-A is not really being transcribed when they knock down ChimA, and they don't really have any protein detectable when they check it by Western blot. So those things are consistent. Now, at this point, they've got a phage that delivers its DNA with proteins, and those are inside of this EPI vesicle. And then DNA starts to get transcribed. And that DNA is getting transcribed. Somehow the mRNA is getting out of the vesicle and being accessed by the ribosome so that it can be translated into protein. And eventually that DNA has to get out of the vesicle into the phage nucleus so it can be replicated and then packaged into new virions. So what they do, the last thing they basically do is look at where does the phage DNA go and where does the vesicle go. They see that by this fluorescence microscopy technique, the phage vesicle goes where the ChimA protein builds the nucleus. It's right next to it. And then they see that it stays there for basically the rest of the time that they're looking. And at the beginning, I mentioned that Gosler does this weird thing where the tubulin-like filaments will rotate the phage nucleus. And they see that the vesicle also rotates as though it is attached to the phage nucleus at a fixed position. And that's kind of where it stops, which is great, but also leaves us with so many questions about how the DNA is, how is the vesicle being made? How does the mRNA get out? How does the DNA get from the vesicle to the nucleus? All kinds of interesting new questions. But the big picture is the phage induces the formation of a vesicle that holds DNA. That DNA is protected from the rest of the cytoplasm, but it is actively transcribed. And eventually that DNA gets transferred into a protein compartment that the phage also makes.
I have questions. So they say somewhere in the paper that they think that the early vesicle is derived from the inner bacterial membrane. Mm-hmm. Is that just a thought, or do they have evidence for that? Do you know?
I have heard them talk about looking, but I haven't seen the evidence yet.
I mean, if that's the case, it makes it sound like during the infection process, the DNA and associated proteins are like budding through the inner membrane. That's a slick trick.
Yes.
Now, there are some phage that actually have lipid in the particle, correct?
Yes.
And I was wondering whether they do anything my first reaction was maybe this phage has lipid in the particle and it delivers this whole thing. Okay, but apparently that's not the case, right?
I haven't seen any proposal like that. That's an interesting idea. You're suggesting maybe the whole DNA in the vesicle is inside the phage head, and the whole thing gets extruded into the cell.
I mean, I doubt it. I think if that were the case, they would have said something about that.
Probably.
And I'm sure they know whether or not there's lipid in the particle. But it made me think. I mean, I was thinking about it because I'm aware of the fact there are some phage that have lipid in them.
Yes.
So my other question, this interests me. They said that there are 2 RNA polymerases, one for transcribing early genes and one for transcribing late genes. Okay, so, and that they are multi-subunit polymerases. So the scheme there would then be that you bring in the early polymerase.
Yep.
And some of the transcription products are going to be for the late polymerase.
Correct. That's right.
Some of the early transcription products, which then will transcribe the late genes, and among the late genes must be the early polymerase that then gets packaged into the virion. I'm wondering if anything's known about the structure of those things in terms of the subunits or anything. I'm wondering if they share subunits, how many subunits they have. I mean, I know this is trivia, but I'm a polymerase guy. Do you know anything about that?
I don't know the answer to that question, but I do not believe that they share any subunits. I think the vRNAP is one protein. Maybe it has a couple of other proteins that work with it, but I think there probably are other proteins that work with it.
Okay.
Now that I say that out loud, because the other ones that I know, they usually use host proteins instead of having another one on their own. But the one that works inside the phage nucleus, the protein compartment, that one definitely has multiple subunits. Sharing? They don't share any subunits.
Do other phages make a phage nucleus besides these Chimaliviridae?
No, not that we know of.
So the
Maybe.
DNA just goes...
The answer, I should say, is we don't know. We only know that the Chamali viruses do it.
2 multi-subunit RNA polymerases carry out transcription.
Okay.
A virion and a non-virion RNA polymerase. Then they say no more.
Fine. So, Rich, this is reminiscent of pox viruses, isn't it?
Absolutely. That's why I'm engaged here. That's partly why I'm engaged.
And later they say that the non-virion RNA polymerase is responsible for expressing late genes, and that's localized within the phage nucleus. So
correct. And that means that the non-virion RNA polymerase and all other proteins that work with it have to be imported into the nucleus compartment, or the nucleus compartment has to be formed around them.
Rich, in a pox virus factory, is it delineated by some kind of membrane or protein?
That's a good question. There are publications that suggest that very early during infection, that the factory is surrounded by some membranes that disappear later on. It's never been clear to me whether that was, air quotes here, deliberate, or whether it's just the growth of this thing in the cytoplasm is pushing membranes aside that then seem to aggregate around the edges. The mature factory. No, actually, you can. I've got some EMs. You can see membrane around mature factories as well. But once again, I don't know if that's a functional thing. It's not as if it's a vesicle that I'm aware of. Or I always thought of it as just the factory just pushing membranes aside and they're accumulating on the periphery. But that's just my prejudice.
So, Jillian, how did the transcripts get out of this vesicle? Do we know?
I haven't seen anybody talk about that yet.
Man, they must make some sort of portal, or
Have to, yeah.
Yeah.
there's going to be a bunch of proteins that get together and make a port to pump this stuff out.
I am almost certain that they must have by now purified these vesicles from the cells to look at what proteins are associated with them to get at that question. I know there's been a lot of effort at how the proteins are getting in and out of the phage nucleus, the protein compartment, the ChimA compartment. I'm sure that there's going to be great effort going into how they get in and out of the lipid compartment, because that's much more like the eukaryotic scenario where you've got mRNAs coming in and out. Maybe you have proteins come in and out. The DNA has to get out at some point, and it's not going to, it has to get out somehow. So,
But still be
is there
protected.
going to be a nuclear pore?
Does assembly go on inside the nucleus? That would make sense, right?
It does not. Assembly
Oh.
occurs in the cytoplasm.
My goodness, so you're at risk there for a moment.
Well,
Oh,
what
wow.
happens is if you look at figure 1, is it in figure 1? In the first figure, they diagram how the phage nucleus has these capsids, assembled capsids docked on the outside of the nucleus. So it's thought that the capsids kind of transit down those tubulin-like filaments
Okay.
to
My
dock
goodness.
on the outside of the nucleus, and then the DNA is pumped into the head, and it's sealed, and then the tail gets added after.
Wow. Okay.
Mhm.
Very cool.
Thank you,
Yeah.
Jolene. So I visited the Polianos lab years ago, 2015, 10 years ago.
Hmm.
And we did TWiV 115. So that was fun. I'm sitting in the lab with the two of them.
Yeah, Kit has your interview video on her lab website.
It was a lot of fun. I'm hearing ribosome a lot. So this is really a nice word, right? Ribosome. It's very, and in fact, Elio Schechter in the link that that Rich put in here at the end, he says, "I've always credited the Model E. That's that big centrifuge they used in the first paper, with in the hands of Stanier, Pardee, and Shackman for the discovery of ribosomes, and Roberts, the senior author, for naming them. It describes them well and has a pleasant sound." So I'm thinking, what did he come up with ribosome? It turns out, so George Palade is credited with first seeing them in the EM. He called them Palade granules, and then other people did more work, and they were called, listen to this, ribonucleoprotein particles of the microsome fraction. That's what they were called for many years. So he took ribo and some, the first and the last parts, and made ribosome.
It's much easier to say. I'm glad they shortened it.
And but ribosome is a really nice name, right? I love it. There's another one that's very similar. That sounds really great, but I can't think of it right now. Okay, we don't have time, unfortunately, for letters, but I do want to read one because it's actionable. This is from Cynthia. As always, thank you for your amazing sharing of science and expertise. I wanted to let you know that many of us who work to strengthen our public health infrastructure to help protect all of us from the bad microbes are growing more alarmed every day. At our most recent Stop TB Tuberculosis USA Coordinating Board meeting, we were made aware of a sign-on letter, gives a link for that, calling out the dangers of the recent executive order acting against gain-of-function research. Suspension of many grants have particularly hit long-standing TB-related experiments at a time when TB cases are increasing in the U.S. and abroad, with many more expected since the dissolving of USAID programs. I immediately knew what was going on with this executive order, since you have discussed many times the moral panic being created around gain-of-function research by adherents to the lab leak hypothesis. I like that word, moral panic. That's really good. It would be wonderful if you let your listeners know about this latest attack on science. It is a great excuse to write another letter to our local papers and to make another call to all our members of Congress. And she gives the number of the Capitol switchboard. I think every American should dial that number right now. It would be
I'm
a
told
denial.
that phone calls are more
Mm-hmm.
effective than other mechanisms of communicating with the government. I guess because people have to answer the phone or something.
I also heard that it's a good idea because usually when you call, you don't actually get to talk to your senator or congressperson, but you talk to an aide and they take notes. And it's good to ask them to repeat back what you asked them to
very
write
good.
down and make sure that it's an accurate reflection of what you called about.
Okay, let's do some picks of the week. Angela, what do you have for us today?
Okay, so my pick is kind of just like a compilation of someone's research that I find really interesting. So Mark Berman, he is at U Chicago, and he does research on the environment and how that can affect our mood in different ways. So it's psychology. It's mostly psychology. And one of these reviews that he published, let me see. The title is The Effective Cognitive and Social Benefits of Interacting with Nature. And so he was also on Huberman's podcast recently, and I have to say I listened to Huberman because I think he's cool. He has cool topics. He has cool guests some of the time, sometimes not so much, but this guy was really cool. And basically they talk about how they've done multiple studies. So this is like with humans, different trials where they take someone, so they randomize people and they either like tell them to go on a walk in a city or go on a walk in nature. Or even they did studies where they would show them a picture of nature or show them a picture of a city during like X amount of time. And then they would even take blood work and then there were different ways to measure their cognitive or social benefits depending on like how they felt. And then they would also with blood work see like spikes in cortisol or decreases in cortisol and things like that. And overall, they showed that being in nature for humans is beneficial. Imagine that. Yes. Imagine. But it's kind of crazy to think about that we have to do studies to show this. But it is true that having like even plants in your house, people that have had plants in their homes or near in their workplace, it has like a calming effect. Or even just listening to nature sounds actually has a calming effect on humans as opposed to coffee shop sounds, which a lot of people like to listen to, but they actually showed that it causes like a decrease in 25% at least of like efficiency while working because your brain is just trying to listen to all of these other voices. It's like when people say there was, he made a good example, like he said this on the podcast. He said, so when students are studying, they all want to listen to music and they don't want to be in silence when they're studying. And then he asked those same students, oh, would you want to listen to that same music while you're taking an exam? And the students were like, absolutely not. And he's like, well, then why is it any different while you're trying to learn when you're studying? So, and one of the hypotheses that they had about nature and why it's so calming on the mind is that nature is created in fractals, like repetitive, recursive, self-similar patterns, like tree branching and leaves and all of these things that are the same over and over again in different ways. And somehow that's soothing to the mind because it's something that is like less cognitively demanding because we've evolved with those things as opposed to right angles. Like there's no right angles in nature. And there's a lot of things that we've constructed that are very foreign to our brains. So it's just cool. If you guys want to click on the links and look into his research.
I've seen some broken rocks with right angles.
Okay, broken rocks, maybe. And I think they're probably like 89.9. Who knows? Yeah, but I thought it was just very, very interesting research. And everyone go for a walk outside.
And so besides having plants in your house, what else? You have to go for a walk. I can't just look at trees out the window.
No,
so they showed that it helps. Exactly. They showed that actually just looking at a tree or a plant does help as opposed to obviously going on a walk is best. And if you're going to go on a walk, they showed even with and without music that if you had music and you were listening to a podcast, the benefits were decreased and that they were increased if you were alone on the walk, because also like mental clarity is best being alone on the walk. So wait, one of the experiments they did, which is a little bit insane, but they took people that were depressed, so people that are clinically depressed, and they let them go on walks, which their hypothesis was that they would ruminate more and they would come back worse. But those that went on walks in the forest, even though they were ruminating, still had benefits from being in the forest, even though they were people with like diagnosed depression, which I thought was very interesting. So
this is really timely for me because the reason I was out the last couple of times is because I spent 10 days in Oregon in the high desert, south of Bend, and my wife's family, we are fortunate, has a house in a resort community there, and it is situated so that it overlooks the Deschutes River through a pine forest. And so you get up in the morning and go out, and it's in the 50s, and you stand on the balcony, and all you can see is the pine trees and the river, and more pine trees across the river, and all you can hear is the occasional chirp, or if a flock of geese goes by, you don't hear any internal combustion engines. It took about a week. And my head was retreaded. Okay, I just sort of rebooted the whole thing because I was pretty wound up when I got there. And I remember I actually commented to my wife. I said, "This is amazing," and I wonder whether what is this? Are we hardwired to connect to nature? And it has this effect, and according to this, yeah. And that makes perfect sense. Isn't it interesting that you don't have to think about it, you just feel it? Okay?
It's true. You do. You walk into a forest and you just take a deep breath and you suddenly feel better.
One of my colleagues thinks about this sometimes, and apparently there's a term for it called forest bathing.
Yes, I also heard of this, which that Dr. Berman doesn't really like that term because he's like, but he did say that, yes, it is like forest bathing where or it's like a form of parasympathetic downregulation, let's call it.
So would a picture of a forest be okay? And you have some music playing too?
He said, so they actually did this study where they put a picture of a forest versus no picture of the forest. And that also had positive benefits.
Okay.
So if you can't get outside, at least looking at the trees, like even in a picture, or listening to like rainforest sounds or birds chirping also calms the mind.
Thinking of millions of people who live in cities where there isn't much greenery, right?
I know.
I wonder what the impact is on them if they can't get to a forest, right?
They've done studies on that too. Apparently, if your window just looks towards even like a patch of grass compared to if your window looks at a concrete wall, it makes a difference in your ability to focus and plan long term and stuff like that.
I got to get a picture of a forest here at the incubator because my windows look out on concrete.
Exactly.
I was going to say, Vincent, you're in luck. You don't even have to go outside. Okay, you can just get some pictures. You're going to be cool.
I don't mind. I went outside to Brooklyn the other day, but it was more concrete. There were no plants at all.
Well, as I was having this epiphany in Oregon, in order to answer for myself the question of whether or not this was hardwired, I was thinking that the control would be somebody who was raised in New York City. Okay? And, if you take them and stick them in Bend, Oregon, are they going to feel relaxed or is it going to make them nervous? Okay. Are they going to be agoraphobic?
But then another question is, would they have agoraphobia or whatever just because they lived in a city, but that's not necessarily their natural state? Is it because their mind is always so full of chaos that when it downregulates so much that there's silence, they don't know what to do with themselves? But it's not that their brain shouldn't naturally be in that state. They've just been so conditioned to being in such chaos, because it literally is chaos. Like listening to a coffee shop and other people's conversations, people listening to that music to try to calm down is a little bit crazy when you think about it. Like listening to clanging plates and people talking as opposed to not wanting to listen to a bird chirping seems a little wild, I think. But anyway.
Thank you very much. That's very interesting.
You're
Rhian,
welcome.
what do you have for us?
I have an essay that I don't exactly remember how I came across it, but this essay was written by three people who are authors of a cell biology textbook that are, or a couple cell biology textbooks that are commonly used, one of whom is Bruce Albert's. And this is an essay called Why Trust Science that explains the process of science, how scientific knowledge is produced, and things like, peer review and iterative going through and trying to test hypotheses and things like that. They are now including this essay in their cell biology textbook to help students kind of understand the process of science, but they've also made it available for free online so that anyone who kind of wants to understand the science process can understand it. There are a lot of nice examples here from different parts of science. Since I've been in the mode of planning some of my courses for fall, I'm kind of thinking about ways that I might use this essay with my students as they're coming into some science classes to really think about the process. I really liked the way that this talked about specifically, thinking about vaccines and public health efforts and how you actually prove that some of these things work. And so I really enjoyed reading it. And I thought it was something I absolutely wanted to share with all of my students. And hopefully it's something that others might want to share as well.
There's a cool figure in this that's sort of a stepwise guide to critically thinking about a process.
Yes.
And deciding whether or not you're going to accept or reject it, which we kind of talk about in a casual way often, but this is very organized. It's
yes,
great.
it is. And right above that part, it's actually using Koch's postulates as an example,
Okay.
but in a more simplified version. And since that's something I think about in a lot of my classes, that's another reason why I particularly liked this, is it used that exact Koch's postulates as an example of the science process.
Kathy, what do you have for us?
Well, first of all, I have something that a listener sent in to me. It was a follow-up on what I mentioned in the Charlie Rice ASV Montreal temperature TWiV, where I talked about the palindrome pattern that 82 Fahrenheit is 28 Celsius and 61 Fahrenheit is 16 Celsius. And then he set to work trying to figure out if there were some others. And he said if you add a 0, and he lists them with the Celsius temperatures first, but if you put a 0 before 4 degrees Celsius, then that could be palindromically 40 degrees Fahrenheit, close enough. And then he also then said if you disregard the 1 in 104 degrees Fahrenheit, then it pairs with 40 degrees Celsius. So it adds at least one more for me, that 04 degrees Celsius is 40 degrees Fahrenheit, and then 16 is 61, and 28 is 82. And other than Rich, I don't think many of us are going to have need of the 104 Fahrenheit conversion to 40 degrees Celsius. But
I had asked you that at ASV, remember? Is there another one? Because you would...
Well, and I said,
decided.
And he agreed that, so it's somewhere around minus 42 Fahrenheit is minus 42 Celsius, or very close.
Yeah.
I don't remember exactly what the numbers are, but, and these are all kind of rounded. I mean, they're not perfect, but yeah. So he picked up on your question and added some more. And then my actual pick is an article I saw about dental floss delivery of vaccines to mice. And so what struck me about it, it was just an interesting, creative way to think outside the box. And the article describes that years ago, an engineer at North Carolina State University who specializes in nanomedicine named Harbinder Gill was reading about gum disease, and he learned that the pockets of gums that are between the sides of your teeth can absorb molecules really well. And so he wondered, could that be used for vaccination? So he and another chemical engineer at Texas Tech tried to floss a mouse. And so they show a picture of it. They had to use like a key ring around the mouse's jaw to hold it open, and then they're trying to floss the mouse. Anyway, so how practical this would be in real life remains to be seen, but I just thought it was an interesting way to think about it. And they first did some stuff with fluorescently labeled protein and showed that they could deliver that to mouse gums, and then they used inactivated flu virus and added that to the floss and could protect mice from flu. So just creative way of thinking about other ways of vaccine delivery. And I can't remember. They described that you could maybe just use dental picks as the way to do this, and maybe you would get these immunizations at your dentist or at some place like that, and that people who really dislike shots would be okay with getting a flossed vaccine.
But wouldn't this be just the same as like sublingual? Because all it is like a highly perfused area, mucosal surface, like your gums.
Well,
Isn't it just?
I think it's just really concentrating it into that specific area. So yeah.
Poor
This
little
reminds
mouse.
me of Frank Zappa moving to Montana to grow dental floss.
Yeah, going to be a dental floss tycoon.
Yep. Dental floss tycoon. Now you can grow it with vaccines in it. That's cool.
So, what do you do for a living? I floss mice. You mouse people there. Can you imagine flossing a mouse? It seems to me the mouse wouldn't really be very receptive to that.
No.
Well, it took
Definitely
two
not.
people to do it. One person to hold the mouse and the other person to do the flossing. If you look at the
So
pictures.
stressful.
Yeah.
Rich, what do you have for us?
I have a book called The Monk in the Garden, alias The Lost and Found Genius of Gregor Mendel, the Father of Genetics. The author is Robin Marantz Hennig. And so a couple of weeks, last time I was on, I think it was, I picked these papers that look at the actual genetics of Mendel's peas by sequencing a bunch of pea genomes. Turns out it was a repick, but it was suggested to me by Kathy. Thank you, Kathy. And I have taught, Mendel's peas before, and it always fascinated me, wondering what was going on in this guy's mind as he was doing this stuff. So, and I've been toying with the idea of reading a biography for a while, but Kathy's turning me on to those papers sort of finally tipped me over the edge. And I picked this particular biography because it won a bunch of prizes and it was written by a science writer. I wanted a kind of a light read. I didn't want to necessarily get too heavy into this. And she does a wonderful job because it's got everything is there. And it was really delightfully written. And it's interesting because it's actually only about half the book is Mendel's biography. So he was, now I'm going to forget all the dates, but he was mid-19th century, 1850s, when he did his experiments. And he was a monk, and he did this stuff in the garden and of the monastery. And those of you who've studied any of this stuff know these experiments. He bred peas and studied the inheritance of different characteristics. What I didn't realize is that, in fact, there were breeding experiments that went on before that, so that the idea of dominance and recessiveness was not entirely novel, okay? They knew that traits could disappear in breeding and then reappear in subsequent generations, but nobody had ever done the quantitative experiments and done the math, okay? And Mendel, by doing a very organized group of studies, came to the conclusion that organisms have two copies of whatever it is. You can call them factors or units. People were mystified by what this could possibly be, but had two copies of whatever it was that for any given characteristic. And Mendel went further and figured out that during fertilization, each parent is contributing only one of those two copies. Okay, so the implication is he had diploid and haploid figured out, but I don't know, who knows what was in his head, okay, because nobody ever attached these things to some physical entity. And he published one paper that was 35 pages long and gave oral presentations of that paper a couple of times, and it was a real yawner. Nobody cared. And so people forgot about it for like 50 years. And interestingly, he became the abbot of the monastery. And so he didn't do his experiments anymore. And when he died and a new abbot took over, they cleaned out his space, including burning all of his notes and everything else. So the only thing that persists of Mendel are some letters that other people received from him and his paper. So we don't really know what was going on in his head. All right. And it was at the turn of the 20th century that his work was rediscovered by several different groups who were making similar observations. And the one story in this book that got me more than anything else was the description of Walter Sutton's experiments. He was a graduate student at Columbia University studying chromosomes, which were, understanding what they looked like and how they behaved was a new discipline at the time. And he'd done some work as an undergraduate looking at spermatogenesis in a grasshopper that he wound up studying in more detail at Columbia. And basically, by studying meiosis and mitosis and looking at the chromosomes, he came to the realization that chromosomes, their behavior, could account entirely for Mendel's observations. He made that connection. And once again, I wonder what was going on in his head. Was that an incredible eureka moment? Who knows? All right. But cool. Fascinating stuff. So I would highly recommend this. Anybody can read this and dig it. And like I said, I'm always interested. We were talking about this with the Meselson-Stahl experiments, right? How do you come up with a method, this kind of stuff? So, what were these guys thinking? And even when it was all done, what were they thinking? Maybe the reason that Mendel's experiments died was that people had no way to conceptualize this, or that was part of the problem. All right, it didn't really stick until we could, have more experiments and associate it with some sort of physical cellular phenomenon. Great stuff.
I mean, it wasn't until the 1940s, that we realized that DNA is it, right?
Yeah.
It's a long time. We had no idea.
Mm-hmm.
So this TWiV is a history lesson.
Jolene, what do you have for us?
Well, I'm tempted to change my pick, but I won't. I'll just mention that I just read a book called Chatter by Ethan Kross, and it talked a lot about this idea of nature helping with the things in your head and the voice that just goes on. And guess I'll leave it at that. But my pick is this website where you can go. It's an interactive map viewer of the stars. The James Webb Space Telescope project that finished surveying a large slice of the sky, actually a slice that's equal to about 3 full moons. And in that slice, they identified over 780,000 galaxies, which is a lot. And it's really cool as you zoom and it loads, and you zoom and it loads, and they're just beautiful stars and galaxies. And they say it effectively lets you time travel billions of years. And I have no other scientific connection. I just love looking at images of the galaxy that make me remember how small everything is on this planet. And there's so much more out there. And it's really pretty.
This is very cool.
This is awesome.
Mm-hmm.
I have an article in Quantum magazine called "At 17, Hannah Cairo Solved a Major Math Mystery." And this is by Kevin Hartnett. And it's a cool story of this woman who disproved a 40-year-old conjecture in math, and it just goes through her story. So she grew up in the Bahamas, and her in Nassau. Her father went there, excuse me, to do work, and so she was homeschooled, and turned out she had an incredible math aptitude. She was advanced at a very early age, and at some point she convinced her parents to send her to Berkeley to take a class, and she was amazing there. And her professor gave her this this problem that had existed in the field, a mathematician, to solve as extra credit, and she solved it and turned the field over on its end. It's so cool that she did this. And now, of course, she's going to go on and have a career in mathematics. It's called the Mizuhata Takeuchi conjecture, considers functions built out of waves whose frequencies lie on a surface. it states that the energy of that function, a measure of how big the function gets, can only be spread out and concentrated in particular patterns. And so she disproved that, and the math field is like blown away. So this is a really cool story of
I'm thinking
anybody could
Goodwill
do it.
Hunting, right?
Yeah,
Yep.
it's true.
This is great. Good for her.
It's so cool.
We have a listener pick from Ruth. Thank you so much for your commitment to science, the scientific method, and service to the public good. Thank you for speaking out the truth about all that's happening to dismantle core institutions that support health and life in the U.S. It's so hard to see this unfold from over here in the U.K. I know it's hard to keep a sense of humor in the face of all this, but I hope you'd enjoy this El Cordova YouTube skit of U.S. federal agencies hanging out in a bunker. Sad, funny, or funny, sad, but hits the nail on the head. Thank you so much, Ruth. At TWiV 1241, you can find the show notes at microbe.tv/TWiV. If you want to send us questions, comments, or pics, we'd love to have your pics. TWiV at microbe.tv. And again, if you enjoy these programs, we'd love your support. Microbe.tv/contribute. Brianne Barker's at Drew University, BioProf Barker on Blue Sky. Thank you, Brianne.
Thanks. I learned a lot.
Kathy Spindler is Professor Emerita, University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
Rich Condit is an Emeritus Professor, University of Florida, Gainesville, currently in Austin, Texas. Thank you, Rich.
Sure enough, always a good time.
Andrea Mingarelli is at McGill University, Immune Vet on Blue Sky. Thank you.
Thank you so much. Go out for a walk in nature if you can this week.
I'll look at some pictures.
Okay, good enough.
I wonder if
We're
pictures
going to
of
send you a
space,
poster, Vincent.
I wonder, like pictures of space, like Jolene's pic would help too, you know. But humans didn't evolve in space, so it probably wouldn't work.
True.
Well, we kind of did.
We
Also
kinda
true.
did.
A long time ago.
Jolene Ramsey's at Texas A&M University, ramseylab.vercel.app. Thank you, Jolene.
Thanks, guys. I enjoyed talking about DNA and phage today.
I enjoyed learning about it. That
You're
was
just
great.
going through a phage.
No, this is not a phage.
I'm Vincent Racaniello. You can find me at microbe.tv. And Microbe TV's on Blue Sky, microbetv.bsky.social. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene Ramsey for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.